At the molecular level, adaptive immunity relies on T and B cell receptors scanning for antigens. The physical site of recognition is the CDR3 loop. The effectiveness of the adaptive immune system depends on the diversity of these T and B cell repertoires. That diversity is primarily determined by the structural variations within the CDR3 region. Chronological time ticks forward uniformly, but biological immune time does not. As we age, the sequence diversity of our immune repertoire degrades. Most existing data on structural immunosenescence comes from European cohorts. Until recently, there was no high-resolution, full-age baseline for non-European populations. Without a precise mathematical baseline for natural aging, it is impossible to accurately quantify how environmental stressors and novel viruses alter the immune trajectory. A 2026 study established a new baseline by mapping the T and B cell repertoires of a Chinese cohort spanning ages 25 to 93. Researchers analyzed this data to quantify natural structural immunosenescence and measure how a severe infection like SARS-CoV-2 alters these metrics. By analyzing these sequence frequencies, researchers built a predictive machine learning clock to assess biological age. The structural parameters of the immune repertoire shift in predictable ways, serving as a biomarker that maps the progression of natural biological aging and post-viral dysfunction. The healthy cohort data reveals that as chronological age increases, the global immune repertoire undergoes a measurable contraction. These charts show the relationship between age and the lengths of CDR3 amino acid sequences. The red bars indicate a negative correlation. Over time, these receptor sequences physically shorten. Alongside shorter sequences, the overall usage of dominant VJ gene combinations steadily declines. This data compares the loss of distinct clonotypes between receptor types. B cell receptors degrade at a rate of 507 clonotypes every 3 years, a steeper drop than the 179 lost by T cell receptors. This contraction is a structural homogenization of the immune arsenal, reducing the body's capacity to recognize novel antigens. A healthy immune system maintains clonal homeostasis, balancing the sizes and proportions of specific clones throughout a lifespan. This Voronoi tree map visualizes a young immune repertoire. The dense small polygons represent a highly diverse, evenly distributed pool of clones. In the older repertoire, aging forces the system into a state dominated by a few massive hyperexpanded red clones at the expense of diversity. To track this decline in diversity, researchers used the Shannon-Wiener index. These low S-curves plot that index across a lifespan. The decline is not linear. There is a distinct inflection point at approximately 60 years of age, where diversity sharply drops. This critical shift from diverse structural plasticity to hyperexpanded rigidity represents a severe mechanical failure point of natural immunosenescence. Researchers then analyzed a follow-up cohort of COVID-19 patients, shifting the focus to acute viral-induced remodeling. These heat maps show similarity coefficients for gene usage. Post-infection, there is a sharp decrease in sequence similarity and altered VDJ gene usage across the cohort. The repertoire remodeling triggered by SARS-CoV-2 closely mirrors the structural parameters of advanced natural aging. These box plots map Shannon-Wiener diversity before and after infection. There is a measurable drop in sequence diversity following the virus. This loss of diversity pairs with a spike in the Gini coefficient as clonal distribution becomes increasingly uneven. Functional annotation shows that these expanded clones specifically map to SARS-CoV-2 and opportunistic pathogens like Mycobacterium tuberculosis. There are age-dependent limitations to this response. Older patients mobilized a significantly smaller fraction of new clones against the infection compared to younger patients. Forcing an already rigid immune system to mount a response against a novel threat exhausts its remaining structural plasticity. Severe SARS-CoV-2 infection does more than trigger an acute inflammatory response. It shifts the immune repertoire further along its natural aging trajectory. Researchers required a mathematical model to unify the data of natural aging with the observations of viral acceleration. They built a light GBM machine learning model to predict biological age based on immune sequence data. The model processes the exact usage frequencies of 55 highly specific VDJ genes and VJ combinations. The primary output is adjusted age diff, which calculates the variance between a patient's chronological age and the biological age of their immune system. When applied to the post-COVID-19 cohort, the model demonstrated a quantifiable increase in adjusted age diff. The structural frequencies of adaptive immune receptors are sufficiently deterministic to serve as a biological clock. Does this microscopic sequence remodeling correspond to a patient's physical health? Higher adjusted age diff values directly correlate with measurable reductions in physical intrinsic capacity. As biological immune age accelerates, physiological metrics decline. Grip strength drops, mobility slows, and oxidative stress markers rise. This sequence-based model connects molecular repertoire remodeling to observable clinical aging. Structural immune shifts provide a quantifiable biomarker for assessing long-term post-viral dysfunction, mapping the biological impact of infection.